Good evening, and welcome to the SMS Research Foundation webinar. My name is Christopher Iannuzzi, and I serve as chair of the Scientific Advisory Panel of the SMS Research Foundation. Thank you for taking the time to be with us. Our hope is that you will find the next hour informative, exciting, and more importantly, inspiring. As most of you already know, today is World SMS Awareness Day, an occasion when those of us immersed in daily life with SMS make a special effort to educate our friends, our family, and our communities about what Smith-Magenis syndrome really is, what it is like for our dear children who persevere every day with their unique personalities and struggles, and equally as important, it is a time to help people understand the complexities that exist for the families of children with SMS and the impact on their lives as well. We can think of no better occasion to announce our new research endeavor. The SMS Research Foundation will fund a $500,000 gene therapy project at the Baylor School of Medicine, the first of its kind in SMS, and tonight you will learn the rationale and details of this project. We'll start with a brief overview of the SMS Research Foundation. Then we'll hear a presentation from Dr. Gianluca Perazzi, a member of our SAP. He has three objectives this evening. The first is to provide a general overview of SMS and the RAI1 gene as it relates to this microdeletion syndrome. The second is to review gene therapy as a concept so we have a better understanding. And the third is to discuss the gene therapy project at Baylor in particular. We'll then have some closing remarks and we'll have some time for questions. Our mission is simple: to support research to improve the knowledge and understanding of Smith-Magenis syndrome so that viable therapeutic options can be developed to improve the quality of life of those with SMS. We have 8 board of directors, all are parents of kids with SMS. Special acknowledgement to Jennifer and Missy, who had the foresight to recognize that we needed a foundation solely dedicated to the science behind SMS. Our scientific advisory panel exists to counsel our board and provides a rigorous review process to ensure that the dollars spent on research are utilized on projects of the highest scientific principle, giving us the best opportunity for success. We, of course, recognize that experiments fail and research direction may change, but it's important to critically review all the options before moving forward, and we have a team of scientists that are dedicated to this process. Since our inception in 2010, we've raised nearly $800,000, and we have proudly supported a number of basic science research projects that have led to a better understanding of the RAI1 gene. The SMS initiative at the Baylor College of Medicine represents our most recent efforts and supports the collaboration of Dr. Sarah Elsey and Dr. James Lupsky, 2 of the leading researchers in SMS. And these grants that we have provided have led to over a dozen peer-reviewed publications and scientific presentations related to SMS and RAI1. You can visit our website, smsresearchfoundation.org, for research updates and to find ways to get involved. So tonight, this webinar will be recorded. We'll distribute it on Facebook, Twitter, and our website. Questions can be asked at any time during the webinar by using the question tab on the right side of your screen. We will answer all the questions at the end of the presentation as time allows, and there will be a short survey, and we kindly request that you take a few minutes to share your impressions with us. Now I'll turn things over to Gianluca, who has been instrumental in making this project a reality, and the SMSRF is grateful for his input and counsel. I will let him introduce himself.
Thank you, Chris. Thank you very much. And good evening, everyone. I'm very pleased to hear there's many people joining us tonight and for this very interesting and fascinating initiative. I'm going to briefly introduce myself. My name is Gianluca Perazzi. I'm a clinical immunologist, and I did my medical studies in Rome, in Italy, where I'm originally from. And I did my PhD in immunology at the Pasteur Institute in Paris. I've been in the US now for more than 7 years, working in drug development for the pharmaceutical industry, Sanofi Genzyme, which means that basically my job is to discover and produce new medications. In addition to that, I happen to be father of Valentina, a very sweet 3-year-old girl with Smith-Magenis syndrome. Soon after my daughter was diagnosed, because of my interest in research, I joined the SMS Research Foundation. I found them on the internet, and I had the pleasure to be appointed scientific advisor by Chris and the board. The first thing that I told Chris in that situation was, I think we really need to tackle the problem to the root. We ought to work in gene therapy. This is a genetic disease, so we have to try to do something to solve the problem. So I'm very happy and pleased to be here today to present this project of SMS and gene therapy. We're going to discuss today, first of all, what is SMS, what are the causes of SMS, and how can we explain the disease. We're then going to review gene therapy, the state-of-the-art with very innovative technology. And then lastly, we're going to discuss the project, gene therapy for SMS. Smith-Magenis syndrome is a very complex genetic disorder. I'm sure many of you, parents connected, know the syndrome better than anyone else. Multiple congenital anomalies caused by either a deletion of the chromosome 17, which include the RAI gene, or a mutation of the RAI gene. There's a very typical facial appearance, intellectual disability. Our kids will be dependent for life. Speech delay, motor delay, narrow behavioral abnormalities, and some of them, I'm sure that you agree, are pretty cute, like the self-hugging, but most of them actually are difficult for us parents, like the self-injury behaviors, the tantrums, the sleep disorder, and many of our kids are also obese. Well, all these are very different features, and there's a different degree of severity however, despite that, all these features tend to be consistently present in all individuals with SMS. And these really represent the signature of SMS. And the current thinking is that these features are all due to the one gene, either the mutation or the deletion of one gene, which is the RAI gene. In addition to these features, there's a number of other less common features, more variable clinical manifestation, all listed here in this slide. Well, not all kids with SMS or individuals with SMS have these features, and some of them are not even typical of SMS, but can be observed in individuals with SMS. Now, the current thinking is that some of these characteristics are probably due to other genes affected and not the RAI gene, since these features are generally seen only in the individuals with the deletion, but not in the individual with the mutation. So going back to the causes of SMS, what is really the genome and what is happening in our kids? Well, the human genome is composed by a total of 46 chromosomes, including 22 pairs plus the chromosome 23, which defines the gender. So XX for female and XY for male. Now, in the individuals with SMS, only one chromosome, the 17, is affected. As you can see, the 17 is actually, it's a pretty small chromosome here. And also only one of these two chromosomes is affected. The other one is actually normal. So if I look more specifically at the chromosome 17, we also find out that the region which is deleted is actually not so big. It's a small region in the small arm of the chromosome here. The deletion is of about 3.7 megabases on average. Some kids are bigger deletions, some kids are smaller deletions, and contains about 80 genes. But they all contain also the RAI gene. So to give you a perspective, the human genome contains over 3 billion bases and over 20,000 genes. So really, there's not so many genes that are impacted, and yet, despite that, you have such a complex syndrome, which is Smith-Magenis. Why? So what happened when a part of the gene is deleted? Well, first of all, we lose some genes, as I mentioned, around 80. Different genes now are next to each other along the chromosome, and before they were not next to each other. Also, the expression on some genes can change because they're not next to each other, and also some of the genes are actually broken through the deletion, causing issues, problems, which are the features, the symptoms of SMS. But most importantly, haploinsufficiency happens. So what is haploinsufficiency? Well, it means basically that only one gene of a copy is present. To be able to understand that, we can look at this picture. On the left-hand side, the chromosome here is a normal chromosome. On the right-hand side, the picture shows a deleted chromosome. So as you can see, in each chromosome, there's a number of genes, which are here letters, and you always have a couple of them, AA, BB, GG, HH. But in the chromosome where there's deletion, then you lose some of these genes, and you only have one gene C, one D, one E, one F. Only one copy remaining. Now, what happens when you have this type of situation is that when one copy is remaining, this copy can compensate for the lacking one in some cases, and therefore you have no consequences. In some other cases, actually, you cannot compensate. And in that situation, you do have a phenotype, you do have a disease. In our specific case, the RAI gene is not able to compensate. And because of that, there is dysfunction. Now, we have certainly many different types of deletions. I'm pretty sure that for those of you who are parents, you know that your deletion of your kid could be larger or smaller than 3.7. Well, here you have a representation on the right-hand side of different individual, and the red represents the deletion. As you can see, different from one individual to the other. Nevertheless, there is a region, there is an area right here in the middle, which is common, and it's critical for all individuals with SMS. In that area, you have the RAI gene, which means that when this gene is missing, you are going to be diagnosed with SMS. You have SMS. So is SMS really due only to one gene, RAI1, or any other genes, or other genes are actually involved? Now, if I zoom it and I show you the picture of the area of the gene which is obviously deleted, you can see this is actually pretty complex. Each of these little names here below, each of them represents a gene, and the RAI1 gene is this one here indicated with an arrow. So this is really the one which is mainly responsible for the syndrome. And why do we know that? But we know that because some individuals, they have a normal genome, but only the RAI gene is altered. So if I zoom again on RAI gene, in this individual, what happened is that only a small portion of the gene is deleted, or maybe it's mutated, it's altered, so that the gene is not normal anymore. It's not encoding for a normal protein anymore. So is really the RAI1 gene the only one playing a role in the disorder, or some other genes plays a role? Well, certainly it really depends on the genetic background of the individual and the expression of some of the other genes as well. So what happened basically is that RAI1 gene caused most of the features of SMS. The haploinsufficiency, the fact that we only have one copy, without compensation from the normal copy, caused the disease. However, other genes that are also deleted, depending on if they're able or not to compensate, can cause other symptoms, like hypotonia, hearing loss, organ anomalies, as well as immune dysfunctions. And that's why not all of these other features are present in all kids or in all individuals with SMS. Having said that, we can summarize this section by saying that SMS is certainly a very complex disease. There are kids with a typical deletion, others with a small deletion or a large deletion, and some of them, they only have the mutation. Nevertheless, they all have the same syndrome, the SMS syndrome. And all of them have an alteration of the RAI gene. So if I replace the missing RAI gene, or I restore the normal levels of RAI gene, can I revert the clinical manifestation of SMS? And most importantly, how do I do that? Well, there's a couple of approaches, therapeutic intervention, that can be used. The first one is gene therapy, where we deliver a gene encoding RAI1 into the central nervous system to increase the expression, to restore the expression to normal levels, and potentially, possibly, restore the function of the central nervous system. In other words, what we can say to layman is that the gene is not there anymore. With gene therapy, we put it back into the central nervous system so it can work again. The second approach is a small molecule approach, in which we can make sure that a drug or a medicine will increase the expression or the function of the RA1 gene. Increase RA1 levels either by activating the expression of the normal allele or increasing the function of the protein which is encoded and present, encoded by the normal allele. In other words, in simple words, either I make the gene, the only copy existing, work more, or I increase the function of the protein encoded by this normal copy, which is remaining in these individuals. Obviously, both these approaches are very interesting and very important. Our interest is to focus on both approaches. But today, we're going to discuss about gene therapy, only the first one of this approach in the gene therapy model for SMS. So gene therapy is a new technology, and I'm sure many of you heard about it, and many of you have questions about it because they think people say that it's something for the future, something that is not really available today or ready today. Well, I can tell you this is wrong. There is a lot of new diseases, and they are currently treated with gene therapy. Gene therapy, what is about, and how we can really classify and how we can really define gene therapy? Well, gene therapy is a transplantation of normal genes into cells in place of missing or defective genes in order to correct genetic disorders. Now, there are two ways to do that. One is called the direct delivery, which means that you package a specific gene into a delivery vehicle and you inject this directly into the patient. The other method is called cell-based delivery, and it's here on the right-hand side of this picture. Basically, you first extract cells from the body, you deliver your gene into these cells first, and then, when these cells are functional, you put them back into the patient. Now, when we say vehicles and we say bringing a gene, injecting a gene into the patients, how do we do that? Well, we use what is called a viral vector for gene delivery. So basically, you use a virus. You modify the virus, you deprive it of all the pathological features, so the virus is not dangerous anymore, and then you include in the virus, you put in the gene of interest that you want to replace into the genome. The virus enters the cell, and the gene can either stably integrate into the host genome, so it becomes part of the chromosome that I showed before, or it can be maintained outside of the genome as a separate piece, but still working and producing the missing protein. there's been very significant progress in gene therapy in the last 20 years. You can see here on the left-hand side, gene therapy is used for vaccine products whose objective is to trigger the immune response against, for example, a pathogen. It's used as gene therapy products to correct the pathological pathways, which would be the case for SMS, for our project. Gene therapy is also used in immuno-oncology to boost the immune system, redirect against the cancer to kill the tumor cells. Now, today we have improved vector designs, which increase safety and efficacy. We have a new development and manufacturing with high yield and purity. We have a very good development of analytics for better quality control of gene therapy, and also improved clinical trial design. And certainly I can say there's a number of emerging clinical success. Some drugs are approved today, are on the market to treat diseases with gene therapy, and many others are coming pretty soon. I'm going to give you the example of one of them, Glibera, which is a very interesting medication. This is produced by Unicure together with an Italian company, Chiesi, for the treatment of familial lipase deficiency. This is a group of rare genetic disorders in which a person lacks a protein needed to break down fat molecules from the digested food. The disorder causes a large amount of fat to build up in the blood. This leads to pain in the muscles, in the bones, also to an enlarged liver and spleen, and also to pancreatitis, which is an inflammation of the pancreas that can be fatal. Well, gene therapy has completely changed the management of these patients, and with the administration of one-time series, up to 60 intramuscular injections in the legs, the kids are completely cured by the disease. This is an example of a very successful approach in gene therapy. As you know, not all of them are so successful, but this is certainly a very successful one. The prevailing view of key opinion leaders today, it's very optimistic. Gene therapy is considered an innovative technology. It can take a long time. It can take up to 10 years to develop a therapy, but it is possible. You have many clinical indications. You can target specific anatomic sites. For example, you can target the central nervous system, which will be the case for SMS. And in some cases, a single injection can provide lifelong treatment. Obviously, some diseases are easier to tackle. For example, when you have an enzymatic deficiency, like the case we saw before, it's easier. Some ophthalmological diseases, but I would say monogenic diseases are certainly the ones that we should be looking at, because since it's only one gene alteration that caused the disease, in theory, if you replace that gene, you're able to treat that disease or at least improve that disease. Well, in the case of SMS, as we learned in the first session of the seminar, RAI gene, one single gene, is responsible for the majority of the features and clinical characteristics. So, can we then apply gene therapy to SMS? Well, first of all, it's important to understand how to study SMS. Well, you can evaluate individuals with the disorder. You can look at the features. You can look at their genes. But also, you can develop animal models to try to mimic the disorder and study the disorder in animals. And finally, you can use blood cells, you can use DNA, RNA, biopsies from individuals with SMS to understand more about the disease. But why we use animal models? Why it's so important? Well, it's important because in the animal model, you can really recreate a disease, you can mimic the disease, and most importantly, you can study the effect of too much of a protein, of a gene product, or too little of that gene problem. And you can only do certain things in an animal because many of these animals, due to the experiments, will die or will suffer from that. And obviously, this is not possible to do in a human. You can use different type of animals. You can use mice, fish, frogs, but mice are generally the most important ones because they're mammals. And believe it or not, mice are genetically, they're not so different from humans. So the SMS mice, so the mouse model that reproduced the syndrome actually exists. It was produced by the Baylor College, and it mimics the human disease. Well, the reasons why it was produced was to determine if too little RAI1 can cause problems in the mice. And as a result, mice have craniofacial defects, they have neurological problems, abnormal strength and walking, seizures, they have a sleep disturbance, and they tend to be obese, which I'm sure it's pretty familiar with many of the symptoms that you see in kids and patients with SMS. On the right-hand side, you have the wild-type mouse here, which is basically the normal mouse. And here on the right, you have the mouse with SMS. As you can see, it tends to be obese, bigger than the normal mouse. So this is the model that we're going to use to study gene therapy and to start the work in gene therapy. And that's why I'm excited and happy to say that this work actually has started. Now, the primary goal of the project is to generate proof of concept with preclinical data that will support the potential testing later on in individuals with SMS. As I told you in the beginning of my talk, I do drug development, so I know very well that for every single medication, you have to start in animals because you have to show what is called proof of concept. What you basically need to do, you need to show that whatever you're doing, it works, you see some benefit in the model, and most importantly, you don't see any safety issue, because it's very important to do something which does not harm the animal. It's safe enough to be translated into a therapeutic intervention. The project will be conducted by the A team. I call this the A team because I truly believe that we have the best people in the world to be able to do this project. Dr. Sarah Elsey at Baylor's College of Medicine is one of the most knowledgeable scientists in SMS. She will be supported scientifically by Dr. San Chung, who's the head of rare disease research at my company, Sanofi Genzyme. Sanofi Genzyme is already doing a number of gene therapy, so they have experience and know-how in gene therapy. The project will be managed by myself and by Dr. Likun Luo, who's also a scientific advisor at the SMS Research Foundation, a very prolific scientist, and a father of a kid with SMS. The project in SMS will be a three-step approach. The approach number one is the following. Due to the large size of the RA1 gene, we need to package the gene into a vector called AAV vector for gene delivery. This is not an easy task because the gene is too large. So the LC lab will identify a minimum RAI gene that still encodes for the functional protein that can be cloned into the vector. I know this is difficult words for some of you who are not familiar with gene therapy, but the way to explain that, if this is the vector in gray, what we are going to do, we're going to make sure the RAI gene here in yellow, it's small enough to fit within this vector. The second step is to construct the recombinant AAV vector, encoding the minimum RAI gene, and then to test that the construct works. So the gene is in the vector, but are we really sure that it is a functional gene? To be able to do that, we'll do some experiment in vitro, infecting multiple cell lines, neuronal cells, or cells coming from individuals with SMS, fibroblasts, to determine the ability of that specific gene in the vector to be functional. So now that we know that the vector is functional and we know that it works properly, then we'll start the step number 3, which is the most exciting step. The step in which we'll actually inject the vector into the mice, SMS mice, and determine the ability to affect functionally and rescue the animals. How are we going to assess success? Well, we're going to look at a number of endpoints in the mice, which including obviously phenotype, circadian rhythm, growth, obesity, to ensure that whatever we are doing is improving the function in the mouse. And most importantly, as I said in the beginning, to ensure that this approach is a safe approach. This project is going to be complex. It is estimated to require at least 2 years to be completed. As of today, and as Chris said in the beginning, SMS Research Foundation will invest about $500,000 in the project, and milestone achievement will trigger payments. We're going to have quarterly meetings with the project management and review committee to ensure and monitor the progress. Obviously, depending on the results and the experimental steps, we may fine-tune or change some of the experiments, and sometimes you need additional experiments to be able to continue the project, which may require, of course, sometimes some additional funding. Research is complex, can always change over time, and that's the challenge that we're going to have. But we are highly confident that we're going to deliver very interesting results. Now, your question may be, this is great, you're doing this in mice, excellent, but what are we going to do with it? After that, what's next? How this translates into human? Well, the way it works is that once you do have a positive proof of concept, you do have those preclinical data, then that's the base, the foundation for developing a medication in human. You can either submit a grant to the NIH, for example, to fund IND-enabling studies, or you can discuss this approach assessing the interest on a number of pharmaceutical companies, either large pharma companies, or you can work with biotechs to develop gene therapy. As I mentioned previously, Sanofi Genzyme already recognized that there is an interest in this indication. Very high unmet medical need with a very complex disease and very emotional involvement for the parents. Obviously, in parallel, I think it's very important to continue working on the small molecule approach to assess if it's possible to reactivate or increase the expression of the RAI gene. This initiative is complex. There's a lot of limitation for a neurodevelopmental disorder like SMS. It's not an easy disease. The complexity comes from the delivery in the central nervous system. Are we able to deliver the right amount of drug? Are we able to reach the right areas of the brain? Are we going to be able to express RAI1 at the right level and at the right time? And most importantly, if everything goes right, and if we do everything right, is the disease reversible? That's obviously a very big question, especially when it comes to adults. I would say it's more likely that the younger you intervene, the better the results are. Is this possible? Well, we really don't know. And what we know is that there's a lot of plasticity of the central nervous system of the brain. So if the study will be positive or not, I think at the end it will depend if you're an optimistic or not. And I am an optimistic. In my opinion, there is the possibility at least to improve the functioning of these individuals, even if when they're adults. And I'm truly convinced that if you intervene as early as the very first years, you can actually improve and reverse a lot of the disease and a lot of the functions. I'd like to conclude this talk by reading the quote of Ellen Johnson Sirleaf, the president of Liberia. She says that the size of your dreams must always exceed your current capacity to achieve them. If your dreams do not scare you, they're not big enough. Well, I had a dream. One day my daughter, Valentina, will understand this presentation. She will understand the genetic causes of her syndrome and how gene therapy helped her from a special child to turn into an exceptional woman. Thank you.
Thank you, Gianluca, for that excellent presentation. I'd like to say now that we invite all SMS organizations worldwide to join this project. We recognize it will take a well-coordinated team approach to achieve our research goals. So please contact us and we will review the potential opportunities to get involved in this groundbreaking project. So now we'll take a few minutes for questions. And Gianluca, the first question is related to age. There's somebody who has an SMS person in their family that's about 28 years old. And, you know, how would gene therapy apply to an older individual? And do you have any thoughts on that?
Right. I think I partially addressed that already in my presentation at the end. This is certainly complex. The nervous system develops over time, over life, and it's true that at the age of 6, 10, maybe, our brain are pretty much formed, maybe continues to grow in the teenagers' years, but then it's pretty much set. So it's very difficult to say for a neurodevelopmental disorder if a late intervention will be effective or not. But what we can say though, is that the structure of the brain and the central nervous system of individual with SMS is normal. If you do an MRI, if you do a CT scan, the brain actually looks the same as a normal brain. You cannot say there's any disease. So if there were some anatomical issues, anatomical abnormalities, I would say the probability of reversibility is very low. But considering that the structure is normal and the size of the brain is normal, if it's really about the connections in the brain, I think there is a good possibility that there is some functions, not all of them, but some function may improve. And for somebody who is SMS, that is familiar with SMS, if I can improve the intellectual ability of 20%, and I can decrease the tantrums of 50%, and I can avoid some of the self-injury or reduce that, I will be extremely happy.
Another question is, when is RAI1 gene normally expressed in an individual? Is it during adolescence? Is it during infancy? Do you have any thoughts about how to answer that?
Right. The gene expression is actually pretty low, and it's expressed all the time. It's expressed at any different moment in life. So the gene is functional and needs to be expressed to be able to exert all the functions. Now, it's important, it will be interesting to see if obviously there is a specific role in RAI gene during the development in uterus. Because if that's the case, if RAI gene is directly involved in the development of the central nervous system and particularly active in uterus, then we should intervene very early, even during the gestational period, to be able to have an effect. However, the fact that the gene RAI is functional also during life means that the lack of the expression in the adults, so the fact is less expressed in adults, also leads to lack of function. Therefore, if we are able to restore that expression to normal level, it's not unlikely that the functioning will be restored.
Are the neuron pathways different in mice than they are in humans?
Yes, of course they are. There's differences between mouse and human. Differences are at different levels. However, what's interesting is that RAI gene is a pretty conserved gene. There's not, as you know, there's some genes that tend to be more conserved. from one mammal to the other, and some other would tend to be less conserved or very different from one mammal to the other. Well, and RNAi is a gene which is actually pretty conserved across different species.
Are there efforts to study the natural history of SMS that will help with clinical studies in the future?
Well, the natural history of SMS and the understanding of that is certainly extremely important. It's very important for that to look into registries and following kids and individuals with SMS and adults with SMS over time to be able to understand disease over time. What is going to be important in our experimental model is to do the experiment at different levels of development of the mouse. So we're going to inject the vectors, in fact, right after birth, later on when the mouse is bigger, so corresponded to a child, and during adolescence for the mouse, and during adulthood for the mouse. In addition, there's also ways today to actually inject vectors in the uterus. So you can really see what is the effect of gene therapy at different stages of intervention. By doing that in the mouse, and we can do that, we will be able to answer to this question. And most importantly, we will be able to answer to the question of, if I do gene therapy to an adult, do I see an improvement? May I see an improvement? Part of those questions can be answered with this type of experiment in mouse.
The next question relates to whether or not you can actually make too much RAI1, and what would that look like, and is there any concerns about that?
Yes, so this is certainly a concern, because the overexpression of RA1 gene causes another syndrome known as Potocki syndrome. So that syndrome, as you may know, is due to the duplication of the RAI gene. So it's the sister syndrome of SMS. So in SMS, you have one gene which is duplicated, and while in the Potocki-Lupski syndrome, you have the gene which is twice expressed as much. So in that situation, you also have a pathological condition. Potocki-Lupski syndrome has some features of SMS, intellectual disability as well, and I would say it's slightly less severe than SMS, but it's still severe. So it's going to be very important that the expression of the gene is maintained or contained within certain levels. And that's going to be obviously one of the challenges that we're going to have with the gene therapy. For that reason, experiments can be done in mice, where actually you can monitor and see the expression of the gene through the vector, and see if it's possible to keep the level of expression within a certain limit. Again, as I mentioned previously, the reason why you do the experiment in mice is not only to assess efficacy, but also to assess safety. And to be able to do that, by transfecting the mice, we will know if an overexpression can be dangerous. I would like to add something else which relates to all the questions that I received so far. All of these questions are extremely important and are actually very spot on. That shows that the high interest of the audience and interest in the disease. But what is really important is that the reasons why you have so many questions and we have so many questions is that we've never done the experiment. That's why it's very important to do the experiment. It's very important to know the answer to this, because whatever we learn from this experiment will help us develop in the future also potentially a small molecule approach. If we learn from this experiment that overexpressing RAI and doing too much of it is not possible, and we learn that gene therapy is not possible, that will make us understand that we need to put our efforts on the small molecule approach and we're working on the small molecule approach in parallel to this approach. With one of the missions on SMS is fund research. Gene therapy is one of them. We're going to look also in the other approach and in the parallel approach, but there's no better way to answer all these questions and to learn more about SMS than doing the actual experiments. In other words, even if the gene therapy itself is not going to be successful in mice, we will learn so much from these experiments that we can definitely increase the knowledge around the disease and help future experiments or future generations find the cure.
The next question is related to whether or not you think there's other gene therapy projects for other syndromes that are further along where we might be able to get some useful clinical information.
Yes, definitely. There is gene therapy further along that are currently in development, including for neurodevelopment syndrome or neuronal syndromes. So gene therapy for SNS is a reality today, and that's why it's very important to leverage the experience of people like Dr. Chang in my company that is working on rare diseases and is working on gene therapy. We have a department in the company which is specialized in neuronal diseases and neurodevelopmental diseases, and that's very important to partner with them as well, exactly to leverage that know-how and leverage that knowledge that can help us in our experiments.
A lot of the questions relate to finances and whether or not the SMS Research Foundation is well funded. And I just would like to say that, we're in a very strong financial position. However, as John Luca had alluded to, gene therapy is unpredictable. Gene therapy is expensive. We know that there will be unexpected expenses that come up throughout this project. And so we could use your